Assalamualaikum and good morning. My name is Norodiyah binti Othman, PhD student from Faculty of Pharmacy UiTM Puncak Alam. Today I will present my research finding with title Anti-proliferative and Apoptosis Induction of Acute Lymphoblastic Leukemia Cell by Madecassoside. For introduction, cancer is one of the leading causes of morbidity and mortality worldwide. In 2020, approximately 19 million cancers were diagnosed, excluding non-melanoma skin cancer, and 9.9 million people died worldwide in male and female. The graph showed top 15 cancers in Malaysia, and leukemia ranked number 9. Leukemia is one type of blood cancer that interferes with the body's ability to make blood by overproduction of abnormal white blood cells. These abnormal white blood cells are called blast or leukemia cells. It was classified by their severity of the disease, either it is acute or chronic, and malignant leukocyte origin, either it is myeloid or lymphoid lineage. This is four main type of leukemia that mostly diagnosed in our population, where the most common is acute lymphoblastic leukemia. Madecassoside is a major bioactive triterpenoid saponin isolated from Centella acetica, and in Malaysia it was called pegaga. The main types of ingredient in Madecassoside is antioxidant and anti-inflammatory, where it can reduce free radicals, inhibit inflammation, and improve skin hydration. These are other triterpene present in Centella acetica, where it can be acetoicoside, madecassic acid, and also acetic acid. Multiple researchers from previous studies have demonstrated the different mechanism of Madecassoside in various diseases. It has shown to give various effects in obesity, age-related macular degeneration, LPS-induced acute lung injury, osteoarthritis, osteoporosis, hepatocellular carcinoma, and many more. However, the potential of Madecassoside as an anti-leukemic agent have not been fully explored. Therefore, the present study was aimed to determine the anti-proliferation rate and inhibition concentration of Madecassoside in acute lymphoblastic leukemia cell, which is CCRF-CEM by MTT assay. The second one is to ascertain the apoptosis rate of Madecassoside in CCRF-CEM cells by flow cytometry, and the third one is to assess the rate of mitochondrial membrane potential (MMP) and reactive oxygen species (ROS) caused by Madecassoside in CCRF-CEM cells by fluorescent assay. This is the methods used to run the MTT assay, which is to see the cell viability. This is for apoptosis assay using a FIT-C NXC-V apoptosis detection kit. This is the method used to see the mitochondrial membrane potential for this study. And the last one is a method used to see the reactive oxygen species in this study. For results, the graph showed that the effect of Madecassoside on cell viability, where cells are exposed to different concentration of Madecassoside for 24 and 48 hours, and ME significantly suppressed almost 20% of cell viability at concentration of 200 micromolar. Inhibition rates were calculated at 48 hours of cell exposure to Madecassoside, and at dose of 40.2 micromolar, it caused 50% of the cell death compared to control, which is untreated and considered as inhibitory dose. Therefore, this concentration was used as for further study. The figure showed the effect of Madecassoside on inducing apoptosis, morphological changes in CCRF-CEM cell at different concentration of Madecassoside, which is IC30, IC50 and IC70, and at different incubation time, which is 24 hours and 48 hours. Two-dimensional dot plots image which propidium iodide is represented versus annexin V fit C, which is AV. RRC, citarabine, is used as a positive control or also as a standard drug. From previous figure, at 24 hours, Madecassoside induced approximately 2%, 3% and 2.86% of early apoptosis cell with a dose at IC30, IC50 and IC70 respectively. The percentage of late apoptosis was significantly increased by dose concentration. At 48 hours, Madecassoside induced approximately also different early apoptosis cell with different dose. The percentage of necrosis were not significantly increased after treatment with Madecassoside at different concentration and incubation time. The graph showed the effects of Madecassoside on MMP level. The changes in MMP was measured quantitatively by using TMRE in control and experimental groups. TMRE gradually penetrates the living cells, stains active mitochondria and exhibit high fluorescent intensity. So this ME exposure increased the intracellular red orange fluorescence as compared to control, indicating the increase of mitochondrial membrane potential. CCCP is used as a positive control in this assay. This graph showed the effects of Madecassoside on ROS level. Exposure of CCRF-CEM cells to TBHP at concentration of 50 micromolar increased the red-orange fluorescence significantly as compared to untreated, an indicator of high level of ROS, which is a positive control. Although there is an increase of fluorescent intensity in cells exposed to ME at different incubation time, which is 0, 1, 2, 3, and 4 hours, there is no significant difference was found in ROS levels between control and ME-treated cells. For discussions, our results showed that Madecassoside inhibit CCRS-CEM leukemia cells in a dose and time-dependent manner. We also found that treatment of Madecassoside on CCRF-CEM leukemia cells significantly increased the population of apoptotic cells as demonstrated by annexin V staining and live or dead assay. Early cellular apoptosis is always accompanied by mitochondrial membrane disruption, resulting in a rapid collapse of the electrochemical gradient. MMP degradation activates downstream caspase, causing cell apoptosis. Meanwhile, ROS formation is an important signal for triggering downstream caspase-mediated apoptosis in many types of cancers. In our present study, the ME-treated cells showed an increase of MMP and ROS production than compared to control, which is untreated. For conclusions, Madecassoside is expected to be another therapeutic option for anti-leukemia because of the promising anti-cancer effects and fewer toxic properties. Although Madecassoside inhibits the viability of leukemia cell by facilitating cell apoptosis and cell necrosis via plasma membrane blabbing and cell shrinkage, further molecular study needs to be done to investigate the underlying mechanism caused by Madecassoside. This is the list of references. That's all. Thank you.